Hi, Year 12. So today I just want you to think about a couple of things to start us off before we start our lesson on synapses today. So what do we need for an action potential to occur? How do action potentials start at the beginning of a neuron, and how do they pass between neurons? So just have a think for a minute, pause the video and then come back. Okay, so hopefully when you're thinking about how an action potential occurs, you've got the idea that you need to get depolarization to reach threshold potential, and then hopefully you remembered the ideas about the opening of sodium channels, voltage gated channels, so we get depolarization to action potential and then repolarization, and you thought about that. How do action potentials start at the beginning of a neuron? Well, hopefully you had some ideas about that from your work on receptors that you did last lesson. So like the Pacinian corpuscle creates these things called generator potentials, which are localized depolarization due to opening of those gated sodium ion channels that change their shape and open due to deformation of the membrane within the Pacinian corpuscle structure. And if enough of that deformation happens and enough sodium ions move in, you then get an action potential. So hopefully your understanding of resting potential and action potential helped you to understand what's going on with the Pacinian corpuscle, and actually that will also help with today's work as well, because what we're going to be focusing on today is how do action potentials pass between neurons. So in other words, how do you then start an action potential in a neuron that's not attached to a receptor. So we're going to be looking at synapses. So we're going to look at diagrams and the structure of synapse. We're going to be looking at neurotransmitters and how they cause action potentials in the next neuron, which are in what are called excitatory synapses. So the synapses you will have covered at GCSE are this type, but at A-level we sort of make a distinction between these things called excitatory synapses. We're going to be looking at the different functions or roles of the synapses. You're also going to think about inhibitory synapses, how they might be different, so what an inhibitory synapse is, and thinking about membrane structure and applying your knowledge of drugs and toxins. Now actually these three here will probably end up being next lesson's content, because there's quite a lot to get through today. So let's think about synapse. So synapsis is joining together, or a junction. If you think about when you did meiosis, you have the chromosome pairs coming together during meiosis 1, in prophase 1 of meiosis, and that's called synapsis. Synapsis just really means coming together. So synapses, there are millions of these in your brain and in your spinal cord, and actually elsewhere in your body. As we've already looked at with the layout of the parasympathetic and sympathetic nervous system, you've got those ganglia, which are actually outside the central nervous system, where you've got junctions between different neurons. You've also actually got these things called neuromuscular junctions, which are the connections between a neuron and a muscle, which are actually similar-ish to a synapse as well. But synapses are these junctions between neurons. And you can see here got a diagrammatic representation and also an electron micrograph showing a similar, showing the neuron here. So this is the presynaptic neuron. Okay, so the one with action potential will be coming down, and this will be the next neuron along the postsynaptic neuron. Okay, the key thing here is there is a gap between the neurons. They're not touching, and the action potential cannot jump between them. This is slightly misleading, this little bit here, but there is definitely a gap, and you need to be aware of that. It's about 20 micrometers wide, and it's too wide for the action potential to sort of trigger depolarization in the next neuron. So all the synapses you have to know about are chemical synapses. Okay, there are actually some electrical synapses where the action potential appears to jump across between neurons, but you do not need to know about them, and they're much rarer. Or for our purposes they are anyway. Okay, so let's have a look at our structure here, and actually what I want you to do is look at this and look at this and go, oh, can I see similar things? So the key thing we have in our presynaptic knob, or presynaptic bulb, depending on what book you read, we have our presynaptic cell surface membrane or plasma membrane. Okay, you can just call it the presynaptic membrane at this stage because it's sort of implied that that's what you're talking about. Okay, we have got vesicles, so little membrane-bound bags containing neurotransmitter chemicals. We've got lots and lots of mitochondria. We've got Golgi apparatus and smooth endoplasmic reticulum here. And then in our postsynaptic neuron, we have got similar sort of structures, especially plenty of mitochondria. Okay, so presynaptic knob, presynaptic membrane, and postsynaptic. Okay, so pre means before, post means after. All right, so postsynaptic membrane here. Okay, on our diagram we can see, look, all these little red dots here. These would be the secretory vesicles. Okay, we've probably got some mitochondria here. Okay, so key features of them just to think about. So they have lots of mitochondria. So why would that be? What do mitochondria do? Well, remember, mitochondria do aerobic respiration, so there's a lot of ATP being generated, so lots of respiration to release energy to produce ATP, because the process of all of the transmission across the synapse and also the action potential itself requires energy, doesn't it? We've got sodium potassium pumps creating resting potential. We've also got generation of vesicles creating molecules, so they're all going to require energy. Okay. We've got lots of smooth endoplasmic reticulum and Golgi. That's because we're creating vesicles with the neurotransmitter in them, and the smooth endoplasmic reticulum is involved in producing these neurotransmitter chemicals. Okay, we've got lots of the neurotransmitter vesicles, and we've got lots of voltage-gated calcium ion channels. So this is a modification of the membrane that's different to the rest of the axon. Okay. Remember, it will be an axon coming down here because it's going away from the cell body, okay, towards the synapse. And what we have in the cell surface membrane here, we have lots of calcium voltage-gated channels as well as our sodium and potassium voltage-gated channels. So they're going to be really important, as we'll see in a moment. In the postsynaptic membrane, we've got lots of mitochondria again, metabolically active. We've got to generate resting potential within the cell, okay. And also there are many sodium ion gated channels, and these are different from the sodium voltage gated channels we have along the rest of the axon. Okay, dendrites, this would be a dendrite here if it's a motor neuron maybe. All right, dendrites, little branches. Okay, and there'll be lots of these sodium gated channels, and they have receptors for the neurotransmitter. So these change shape because of a chemical binding to them. So they're chemically gated rather than voltage gated. So that is a key difference in that postsynaptic membrane here, is they have these channels which have also got receptor sites for the neurotransmitter. So if we're looking here, we can see this is a diagrammatic representation of the structure of the gated channels. Okay, here we've got it in 3 dimensions and cut through. Here's our phospholipid bilayer. So the transmembrane is transmembrane channel protein, and it's made of 5 polypeptide subunits, and 2 of them, the 2 alpha subunits, you don't need to worry about that, have acetylcholine binding sites. So we're going to just talk about acetylcholine when we talk about neurotransmitter, or acetylcholine, depending on how you want to say it. But there are lots of different neurotransmitters, and different synapses will have different receptors for different neurotransmitters, and as we'll see later on, maybe even different channels. Okay. Now, if we look here at the structure, there's actually some negatively charged regions here, and when the channel is closed, you can see all those negatively charged regions are pushed close together, and they're going to be important for the sodium ions be able to pass through. Okay. Because remember, sodium ions are positively charged, so they'll be attracted and go through. But when it's in its closed shape, they can't get through. And when the neurotransmitter binds, they change shape, and then the sodium ions can get through. Please ignore this one saying potassium ion. So the binding site for the neurotransmitter, when the neurotransmitter binds, it causes a change in shape of the protein. Remember, this links nicely into your foundation biology work on protein structure. Remember, if things bind to proteins, they often change shape. So we're thinking of the tertiary structure of our protein changing, so flipping from one shape to another when the neurotransmitter is bound. And in this case, it causes the sodium ion channels to open. So you've got a worksheet with a sort of blank, slightly different looking diagram of a two neurons, the presynaptic and postsynaptic neuron. I want you to annotate the worksheet with as many structures as you can. If you want to do a little sketch of this diagram, you can. You're not going to be expected to draw out a receptor channel protein in the postsynaptic membrane, but at least be aware that they exist and how they work. So label and then annotate features from the last two slides onto that worksheet. Give yourself sort of, I don't know, 5 to 10 minutes on that. Pause the video here. Okay, so what we're going to look at now, I'm going to talk you through the stages that occur at the synapse with a couple of little diagrams here, and then you are going to have a go at doing your own bullet points, and obviously you can use the textbook and things like that as you go through. So I'll talk you through and try and make it as clear as possible. So the key thing that happens when an action potential passes down the axon and reaches the presynaptic knob, the presynaptic membrane will depolarize. Exactly as normal, just going to action potential. And that depolarization will cause calcium voltage gated channels to open, and calcium ions will move into the presynaptic cell, okay, to the presynaptic neuron. Now, calcium ions are really important in generation of movement. So when you do muscles, you'll see these ions are really important again, and they will cause movement of the neurotransmitter vesicles towards the cell surface membrane of the presynaptic neuron. And then we get exocytosis of the neurotransmitter chemical, because the vesicles will fuse with the membrane and release the neurotransmitter chemical into the synaptic cleft. Okay, the neurotransmitter will diffuse across the synaptic cleft and bind to the receptor chemical gated sodium ion channels on the postsynaptic membrane and cause those sodium chemical gated channels, sodium ion chemical gated channels to open. This will mean that sodium will move into the axon exactly the same as if normal depolarization was occurring, but here we've got the chemical gated opening of the sodium channels and some sodium ions will move in. Okay, now it says here that action potential will occur. There's a key thing that happens here is, like with the receptors, you get a generator potential. In other words, we get a depolarization, and it is called an excitatory postsynaptic potential, or an EPSP, excitatory postsynaptic potential. Okay, so in other words, it depolarizes. Now, you have to get enough depolarization here to reach threshold, and then you will get an action potential. So you may or you may not, depending on how much neurotransmitter has been released within a certain time frame. Okay, and I'll talk to you more about this in a moment when we look at roles of the synapses. Okay, so that's literally basically it. So action potential reaches the presynaptic membrane and the presynaptic knob depolarizes. Calcium chemical gated channels open and calcium ions diffuse or move into the presynaptic neuron. That causes vesicles to move or be moved towards the cell surface membrane of the presynaptic neuron, and they fuse with the presynaptic membrane and release the neurotransmitter by exocytosis, which diffuses across the synaptic cleft, binds to the sodium chemical gated channel proteins, which change shape and open. Sodium ions then move into the postsynaptic neuron across the postsynaptic membrane, causing the postsynaptic membrane to start to depolarize or create a excitatory postsynaptic potential. And if that reaches threshold potential, you get an action potential. Now, key thing is, what we don't want to do is keep the neurotransmitter bound to the channel protein receptor sites permanently, because otherwise the sodium channels would remain open and you'd continuously get action potentials. This membrane would continuously and permanently depolarize. So in the synaptic cleft there is an enzyme, and the enzyme in this case, because we're using acetylcholine, is called acetylcholine esterase. And acetylcholine esterase breaks, hydrolyzes the acetylcholine into acetate or ethanoic acid and choline. So acetate and choline are then recycled, so reabsorbed by diffusion into the presynaptic membrane, and then using energy from ATP and processing, they're reformed back into acetylcholine and repackaged into vesicles, ready to go again. So the whole lot's continuously recycled. But as soon as the acetylcholine is hydrolyzed by acetylcholine esterase, the sodium channel closes. So it prevents any further depolarization. So this is happening quite rapidly. So there's a constant sort of battle between the amount of neurotransmitter being released and it being broken down. Okay, and this is why you have to have enough neurotransmitter released within a period of time to get enough depolarization to reach threshold. The excitatory postsynaptic potentials have to sum up in order to reach threshold, and I'll talk to you about that more later on. Okay, so there's an overall review summary. So what I'd like you to do now is try and write your own bullet points of the events at the excitatory synapse. So give yourself, I don't know, seven minutes maximum to write these out. So maybe have a look back at the stages, reading the book, close your book, and then try and write your bullet points out without recourse to the videos again. So copying, remember, is not useful. Try and do it from what you can remember. Okay, give yourself 7 minutes. Okay, so hopefully you got some ideas like this. So the action potential reaches the presynaptic knob and the presynaptic membrane depolarizes. Voltage gated calcium ion channels open and calcium ions diffuse into the presynaptic knob. This causes vesicles containing the neurotransmitter to move towards the presynaptic membrane and fuse with it. So we get exocytosis of the neurotransmitter, which diffuses across the synaptic cleft. Neurotransmitter binds the receptor sites on the sodium chemical gated channels in the postsynaptic membrane. So the sodium ion channel open and sodium ions diffuse across the postsynaptic membrane into the postsynaptic neuron, creating our EPSP. If we get enough PSPs combining, we reach threshold, and then we get an action potential, which to then obviously propagate down the axon or dendron, if it's a, depending on what it is, but if it's a motor neuron, through the dendrites down to the axon, as normal. I don't know why I've put a capital A there. And then we've got our hydrolysis or breakdown of the acetylcholine by acetylcholine esterase, and it's recycling back into the presynaptic neuron. So it might be a good idea just to spend a bit of time making sure you've got these key ideas in your bullet points. So hopefully you wrote out the bullet points and left some space, and then you can go and have a look at that. Okay, let's now think about the roles of the synapse. So what is it there for? So key things. You can have what is called divergence and convergence of the neurons. Okay, so you can have one neuron making synapses with a number of other neurons. Okay, so we're going to multiple postsynaptic neurons, so the impulse can be spread to others. And remember, because it's digital, if we get the action potential going down here and it splits, we get the same action potential there as there. There's no loss in the signal because it's just propagating along. Okay, and these would be the dendrites going to different synapses. So then we can get action potentials in multiple neurons on the postsynaptic side, so we can diverge. But equally, we can also have convergence. So we could have receptors or something like that coming from, and obviously these diagrams are a little bit simplified, multiple different neurons and actually ending up synapsing with a single neuron here. And as we'll see later on, actually some of these neurons might have differing effects on this postsynaptic neuron. So we can have convergence, okay? And actually convergence is also important in terms of generating a big enough excitatory postsynaptic potential to get action potential. I'll talk about that later on, because you can have a few action potentials running down each one, and they could release enough neurotransmitter over the space of this neuron from different spatially, if you like, to sum up to get an action potential. Okay. Synapses ensure that the direction of transmission of an action potential only goes in one direction, and this is really, really important, that the action potentials only ever run in one direction. Remember, we've already talked about that in terms of propagation, but sometimes something could happen to trigger an action potential within the middle of an axon, just by chance. Something weird could happen, okay? And obviously then it would propagate in both directions, but the key thing is it would only be transmitted forwards in the correct direction, because going back the other way it would not be able to be transmitted here, because this would be effectively the postsynaptic neuron if there was another neuron here. Okay, so key thing, they ensure one-way transmission. You want to think about, well, how does the structure of the neuron ensure that? How does the structure of the synapse and the presynaptic membrane and the postsynaptic membrane, how does that ensure there's only unidirectional transmission? They act as a resistor, or they filter out low-level stimulation. Okay, I've already talked to you about the idea that you have to reach threshold. So if you only get, say, got maybe a very low frequency of action potentials, or a very low level stimulus, or something triggering this one to just send a couple of action potentials down, a second, not very many, you're not going to get very much neurotransmitter released, and that excitatory postsynaptic potential won't reach threshold, because remember, that postsynaptic membrane has still got all the other features for generating resting potential, so it will just restore the depolarization back to resting potential if it doesn't depolarize quickly enough to threshold. So it can filter out low-level stimuli. Okay. However, they can also amplify them. As I've already mentioned, you can have this idea of summation. So you might get a low-level stimulation of this one, and a low-level stimulation of this one, and this one, and this one. So only a few action potentials coming down a number of neurons, but that could be enough to spatially sum up here and reach threshold in our postsynaptic membrane. So our excitatory postsynaptic potentials could add up and sum, okay, to get action potential in our next one. So it can also work as an amplifier in terms of what we call spatial summation. Okay. Synapses do get tired or fatigued or habituated. Okay, if you think about what's happening when you're releasing neurotransmitters across the synaptic cleft, eventually you will run out of neurotransmitter. Okay, and you've not got enough to send more across, because it takes time to remake it. And this is what happens with your synapses, and it's why, for example, when you first put your clothes on, you feel them, but after a while you stop feeling the clothes on your body unless you move, and then you get new stimulation. Okay. Why you don't notice a smell in a room after a period of time, because the receptors in your nose and the synapses associated with them become fatigued. Okay, there's a nice little activity you could try at home. Actually, just be careful if you've got three washing up bowls or 3 buckets or something like that, and if you put hot as you can handle hot water in one bucket, really ice cold water in another, and sort of tepid water in the third one. If you put one hand in the really hot water and another hand in the really cold water, and hold them there, okay, for about 30 seconds, and then take them both out of that and put them into the tepid water, see how your hands feel, because you have 2 different types of receptor in your hands. You have hot receptors and cold ones, and which ones will be being stimulated in which of the 2 buckets, hot or cold, and then what happens to how you feel the temperature of the water when you put them into the tepid water is quite interesting and weird. It's also similar to the idea when you first get into a bath, it might feel really, really hot, but if you get used to it, and similarly if you jump into the swimming pool, it feels cold initially, but eventually you acclimatise to it because the temperature receptors in your skin become fatigued. So that's a useful little thing. It's really important in terms of creating learning and memory. Okay, how you learn things is about creating synapses, connections between your neurons. This is why we want you to keep practicing and trying to recall things, because the more you try to retrieve and use those synapses, the stronger those connections become. They make more connections, okay, because they are a junction box, a connection point. Okay, so that junction box really links to divergence and convergence. So, be a good idea now to bullet point these ideas, maybe have a look in your textbook and write any extra notes you want to do on this. And then in the next couple of slides there's some questions to get you thinking ready for the next lesson. So, what I want you to do is explain why the structure of the presynaptic and postsynaptic neuron ensure one-way transmission. Explain what would happen if the neurotransmitter was not broken down by the enzymes in the synaptic cleft. Okay, and compare temporal and spatial summation. How are they similar to each other and how are they different? All right, so you might need to do a bit of reading about that. I've talked to you a little bit about it already. And then when you've had to think about those questions, really have a think about this one. So we've already talked about this idea of neurons can converge on each other, but they actually might have different effects on the postsynaptic membrane. So we've looked at the excitatory synapses where we get an excitatory postsynaptic potential, but some synapses are what we call inhibitory. So they cause an inhibitory postsynaptic potential. So the neurotransmitter being released from the presynaptic neuron will actually result in a reduction in the rate of action potential, prevent action potentials being generated in the postsynaptic neuron. So what do we mean by the term inhibitory postsynaptic potential? How is that different to an excitatory one? And what happens at these synapses when neurotransmitter is released, and how might the postsynaptic membrane structure be different? You can answer that all together because actually it sort of links together. Okay, so have a think about those questions and we'll come back to this next lesson, and then we're going to spend a lot more time next lesson looking at application of this knowledge and understanding of synapses. Okay, thanks.